Okay, good afternoon. Let me welcome everyone to this session today. We really do look forward to this session by Dr. Burt. I will first uh, like to have the land acknowledgement put up, and let me start by acknowledging the land in which Sick Kids operates. For thousands of years, it has been the traditional land of the Huron-Wendat and Petun First Nations, the Seneca, and more recently, the Mississaugas of the Credit River. Today, Toronto is home to Indigenous peoples from across Turtle Island, and Sick Kids is committed to working toward new relationships that include First Nations, Inuit, and Métis peoples, and is grateful for the opportunity to share this land in caring for children and their families. Now, before introducing Dr. Burt, let me mention a few housekeeping items. First, to remind you that the session is being recorded today. So we thank our IT team for that. And then also to mention that near the end, there will be an evaluation, a link for an evaluation that will be put in the chat. So you can please complete that. I'll remind you near the end as well. And then also to give an idea of the next session, November 11th is a special pediatric focused workshop that addresses practical issues related to pediatric vaccines. And we are developing this in close collaboration with the Black Physicians Association of Ontario. So we thank them in that regard. So let me introduce to you Dr. David Burt, who's an immunologist with more than 30 years experience leading research teams in universities and in the vaccine industry, as this relates to the development of vaccines against infectious diseases, including influenza and SARS-1. Now, he was a research fellow at the National Institute for Medical Research in Mill Hill, London, England, and received his PhD in immunology from the University of Birmingham in the UK. He has published over 40 peer-reviewed articles and has his name as a co-inventor on 7 patents. David received the Harry Jerome Award for Health Sciences in 2006 for his dedication to his field and promoting the field of science. He is president of DS Burt, an R&D consulting organization that offers independent consulting advice in biotechnology. And he's a member of the City of Toronto's Black Scientist Task Force on Vaccine Equity, which, as you recall, has a mandate to engage communities that are most impacted by the pandemic, as this relates to COVID-19 and vaccines. So without further ado, join me in welcoming Dr. David Burt. David, over to you.
Okay, thank you so much, Upton, and thank you for inviting me to participate in this really important series of seminars that have been organized. I must say that despite my 30 years in the vaccine industry, I don't think I've enjoyed talking about immunology and vaccines as much as I have in the last year or so at a sort of grassroots level. And it's been really fulfilling, and I'm really proud and really blessed to be part of the City of Toronto Task Force on Vaccine Equity, the Black Task Force on Vaccine Equity. Without further ado, I will share my screen, and actually I pressed the wrong button there. Share my screen. Here it is. Okay. Let me see. So I'll just. Okay, there we go. Okay, so today I've been asked to speak on COVID-19 vaccines, the types of vaccines, how they work, efficacy and effectiveness. And I'm sure that you're all aware that we're in the middle of a pandemic that is bringing new challenges every day, new information, especially in relation to the emergence of new variants. And it's within this framework that the vaccines that have been developed have to work and are being used. And I'll spend some time during this presentation talking about how these vaccines are performing in this ever-evolving situation of the pandemic. And hopefully, I'll be able to transmit the idea that despite a lot of misinformation that's circulating, you'll see that the vaccines are very effective at preventing severe disease, hospitalization, and death, even against the new variants that are emerging. Okay, so the overview will be, I'll be talking about the role of the vaccines in fighting COVID-19 pandemic in general. The vaccines have been authorized or approved in Canada, the mode of action, the vaccine efficacy and effectiveness against the COVID-19 virus, and specifically against the variant strains, and just a bit of information about the new COVID-19 vaccines that are under development. And this slide is just to show that the vaccines that we have now, highlighted at the bottom here in 2020, especially those of Pfizer and Moderna, are a legacy of a few hundred years of vaccine development and technology, starting in the 1700s with the variolation, smallpox variolation that was occurring in the Middle East and China. And I've introduced Onesimus, who was a black slave who introduced the idea of variolation against smallpox to the North Americas. And we can look at the timeline of the vaccine development process, going from smallpox in the 1700s to polio in the 1950s. We have vaccines against Ebola, 2019, all the way to 2020, where we have the mRNA vaccines. And throughout this process, we see that technology has improved, and we've gone from using very crude inoculums for vaccines to very highly purified and defined recombinant protein vaccines. So it's just to let you know that this series of vaccines that we have now against COVID-19 have a history, and we'll talk about that in a little while in more detail. I don't think I need to really provide much evidence to show that vaccines have been effective throughout history. And diseases like measles, pertussis, mumps, rubella, smallpox, polio, after the introduction of vaccines, the incidence of these diseases have been reduced in many cases by more than 99%. So vaccines work. Where do the COVID-19 vaccines come in our attempts to deal with this pandemic? And I like this, this schematic here, showing the different types of interventions, protective measures that we have, as being slices of Swiss cheese. So wearing masks is a slice, physical distancing, proper ventilation, quarantine and isolation, and vaccines. And you can see that these slices have holes in, which suggests that independently, none of these methods of protection are perfect, and viruses can evade mask wearing if we don't wear the masks properly, if we don't isolate properly. And even vaccine that is 90% effective is not totally effective for everyone. And independently, none of these measures are perfect, but together they can greatly reduce our ability to be infected and to transmit virus. And we believe that vaccines are going to be the final piece of the puzzle when enough of the population have been vaccinated. But as you'll see through this presentation, even with very effective vaccines against the original strain, with the emergence of variants, we may still have to utilize many of these protective measures in addition to being fully vaccinated until the population has been fully vaccinated because of variants that are emerging. So COVID-19, as you know, is caused by SARS-CoV-2, a coronavirus, and this is an RNA virus, and probably the most important target for a vaccine is the spike protein, because this interacts with the ACE2 receptor on cells in the upper and lower respiratory tract, and in fact, in various parts of the body. The virus, like most viruses, hijacks the cell's machinery. The RNA is translated into protein. The various proteins of the virus are assembled and the virus is released. But we have an immune system and we have cells that can recognize the virus and present epitopes or parts of the virus to B cells and T cells, which are the effector mechanisms of the immune response against the virus. Now, in terms of coronavirus vaccines, there are hundreds of vaccines in development at different stages. And we know that despite the pervasive belief that a vaccine was developed rapidly, and we took that vaccine because it worked, and maybe it's not the best vaccine. If you look at this, 8 vaccines that were under development were abandoned after the trials because they didn't work. And some of these were developed by some of the real major players in the vaccine industry. So it wasn't a slam dunk that a vaccine would be developed. And in fact, it was very remarkable that we have vaccines that were developed so quickly. But we'll go into that in a bit later. And there are various types of vaccines that are under development against the coronavirus. Most of them are trying to develop antibodies, T cells against the spike protein. And that can be achieved in a number of ways by delivering the whole virus in an inactivated form, maybe formalin inactivated, or by taking the spike protein itself and using that as an immunogen. Sometimes, depending on how the protein is produced, the proteins can self-assemble into virus-like particles. They're very effective. Other types of vaccines, especially DNA vaccines, utilize a viral vector to deliver the DNA into the cell. And of course, we have the RNA vaccines. And we'll go into some of these in a bit more detail. And these are some of the vaccines that have been approved, authorized, or almost approved in terms of Novavax for use in the fight against COVID-19. The first set of vaccines are the genetic vaccines or nucleic acid vaccines. And here we're talking about the mRNA vaccines from Pfizer, BioNTech, and Moderna. And these vaccines work. And initially, a lot of work has been done over the last few decades trying to use mRNA, DNA to deliver them into cells so that they can induce immune responses. And technology has been developed over the last decade or so, showing that you could actually encapsulate mRNA in fatty lipid bubbles. And these are able to interact with the membranes because the membranes are made of lipid of our cells. And these vaccines enable the RNA to interact with the ribosome in the cytoplasm. There's translation of the RNA into amino acids. And finally, you get the spike protein. And this is the protein that is recognized by the immune system. And one of the advantages of this type of technology is that it's not live. So there's no live pathogens. So that reduces the safety issues and reactogenicity that could take place. It's a very flexible way of designing a vaccine. And it's almost like you can plug and play this type of technology, and it's well characterized. The RNA does not integrate into the host genome. And once the RNA is actually translated, it's degraded and removed from the cell. Potent immune responses are produced, and the RNA itself is an adjuvant. I think it's TLR7 or TLR, yeah, I think it's TLR7 or TLR6, but there are receptors on cells that recognize RNA and can stimulate and boost the immune response. So these are self-adjuvating. There's no need to have any adjuvants included in these formulations. And they can be manufactured very easily. There's no need for cell culture or growing up a virus, and can be scaled rather rapidly into large capacity. So these vaccines are very efficiently made and induce a very efficient immune response. What's in them? Well, you have the active ingredient, which is the mRNA. This is an example from the Pfizer-BioNTech. We have the lipids. There are 4 different fatty acid molecules that are involved in protecting the RNA and also enabling the package to be delivered into the cells. The RNA lipid package is cryopreserved because they need to be stored at low temperatures. And this is by adding sugar. And then we have salts that make sure that the pH or the salt composition of the vaccine is the same and matches that in our bodies. So it's a very simple formulation. There are no ingredients, animal ingredients, no preservatives involved in these vaccines. And these vaccines, people ask, how come they were developed so rapidly? We know that BioNTech scientists, that Dr. Sayan and Tereshi have been working on RNA vaccines for cancer and later on for infectious diseases. And perhaps the most significant development was that from the National Institutes of Health, Dr. Kizzy Corbett and Barney Graham, whose teams actually were able to develop the technology using the Middle Eastern Respiratory Syndrome virus as a model. And they were working on this other coronavirus, the Middle Eastern Respiratory Syndrome virus, for a number of years prior to coronavirus COVID occurring. And they were able to rapidly apply the technology to the coronavirus, the technology that they developed for the MERS. And also, this team was also involved in developing monoclonal antibodies, the first monoclonal antibodies that could be used as treatments against COVID. And this was done by actually recognizing that the actual molecule, the S protein, it's like it has a tendency to change its conformation when it interacts with a cell. And in order to use it as a vaccine and to make antibodies for monoclonal antibody treatments, they had to modify it so that they fix the structure so it wouldn't change its conformation. And this was key to developing the vaccines. Most of the protein and mRNA, DNA vaccines are using either the protein or the recipe of the protein that fixes the protein in the conformation that's necessary. And this is a team that's really enabled this to happen. And so we find that work on the MERS-COVID coronavirus between 2013 and 2019, published in key scientific journals, paved the way when the SARS-CoV-2 virus came along at the end of 2019. They were able to quickly, once the sequence was published, quickly adapt their system, and the NIH team and Moderna worked together. And we know that once a formulation was evaluated, it didn't take very long for that material to end up in clinical trials. And we see that we're talking about less than 3 months between the first outbreak, knowing what the sequence was, and developing a vaccine or producing a candidate vaccine that went into phase 1 clinical trials. And that is because of the work that was previously done. And also, of course, because of the heavy support, governmental support in terms of dollars and research capabilities that were given. The other class of viral COVID vaccines that I'm going to talk about are the viral vector vaccines. And these are the vaccines that AstraZeneca and Janssen produce. And if you look at the way that a cell produces protein, the DNA code is transcribed into mRNA, and this mRNA goes into the cytoplasm, it's translated into the protein. And Pfizer and Moderna, their vaccines basically start at this level of the pathway. AstraZeneca and Janssen start at the DNA level, but they all end up producing the same protein in the cell that is recognized by the immune system. And this is an example of the J&J vaccine. The actual delivery vehicle or the vector used is an adenovirus. This is a harmless virus. It's been modified so it doesn't infect the individual, but it can take the DNA into the cell and induce the immune response. And again, the development of this was really rapid. 3 months after the sequence was available for the SARS-CoV-2 S protein, there was material that was ready to be put into clinical studies. And again, this vaccine is very simple. We have the adenovirus vector containing the DNA sequence for the CoV-2 spike protein, and other ingredients which are basically common ingredients that are found in vaccines. Again, no preservatives, no other components that aren't necessary to enable this virus-based vaccine to enter the cell and produce an immune response. The phase 1 trials were done to identify and to show that the vaccines were safe. and also to, in many cases, to identify a dose that could be given, and in other cases to identify which type or variation of the technology should be used. And as you can see, this clinical trials, which are often combination of phase 2 and phase 3, included tens of thousands of individuals that were recruited extremely rapidly. Again, this was a pandemic, a lot of interest in volunteering, and representing a good cross-section of ethnicities and races. For instance, most of these studies had at least 10% of the subjects were Black, and often 20% plus Hispanic and Latinos. So there was a good representation, and most of these studies started in the summer or the fall of last year. And this is just a slide showing the efficacy of these vaccines as a result of the clinical studies. And the clinical studies, efficacy was basically determined based on the ability of the vaccines to protect against laboratory confirmed COVID-19 disease. And you can see efficacy ranging from in a mid 90% range for the mRNA vaccines, for the DNA vaccines a bit lower. But again, these studies were done at different times. The mRNA vaccines were the first to be developed, and they were developed under circumstances where there was more of the original strains of the virus circulating. By the time the Johnson & Johnson and AstraZeneca studies were being done, the vaccines were being developed and being tested in the trials when other variants were developing. So we shouldn't take too much, make too much focus on the percentages per se, because as we'll see, most of these vaccines do what vaccines are supposed to do, and that's protect against disease, hospitalization, and death, severe disease, hospitalization, and death. And we found, and it was found that in real life use, these vaccines were as effective in the field as they were in the clinical studies. And the Israel study using the Pfizer mRNA vaccine showed, in millions of individuals that this vaccine was 97% effective, very similar to what was found in the clinical studies. But we also found from the Israeli studies that after about 8 to 9 months of being very effective, these vaccines seem to lose some of their effectiveness. And this raised a red flag. Was this because of the Delta variant circulating, which was occurring after 8 to 9 months in Israel? Or was it due to the fact that the virus, the vaccines were becoming less effective? This was a study from the UK showing the effectiveness of the vaccines in the UK against the Alpha and Delta variants. So the Alpha in blue, the Delta in purple, after 1 dose and 2 dose. And as you can see, and this was a time, this was I think between April and May, a time when the Delta was only 22% of the circulating strains in the UK. And you can see 1 dose wasn't as effective as 2 doses. 2 doses were required to give really optimal protection. But there's already a hint that there was a difference between the ability to protect against Alpha versus Delta, especially after 1 dose. And maybe not significant, but there was a hint that something was occurring there. And studies in the US looking at frontline workers also hinted that the vaccine efficacy before Delta was around 91% and dropped to about 66% when Delta became prominent. So it seemed that Delta was having an impact. And this is a meta-analysis that was reported in October, early October, looking at different vaccines, looking at different clinical studies where they looked at vaccine efficacy against Delta, and comparing, let me move this out of the way, comparing vaccinated and unvaccinated individuals. And what was seen was that the unvaccinated individuals had a higher percentage of deaths, as you might expect. Also, they had a higher percentage of moderate infections versus the vaccinated. But still, what was concerning was that there seemed to be what was known as this breakthrough infections, where the vaccinated were showing significant amounts of infection, mild cases, sometimes asymptomatic. That was not a lot of difference to what was seen with the non-vaccinated. And this table shows that while infection was more common now in the vaccinated against Delta, severe infection was still being protected against, and death was being protected against for various vaccines under development. And in this study from the US, from the CDC, we see this line segregates the time when up to the beginning of July, when the Delta variant wasn't dominant, versus when the Delta variant became more dominant. It was more than 50% of the circulating strains. And you can see the cases of in unvaccinated individuals were dropping, but started to rise with the rise in Delta. And even in the vaccinated, there was a rise when the Delta, the rise in infections or positives, lab positive individuals when the Delta arose. A rise in hospitalizations for the non-vaccinated, but the hospitalization rate was remaining quite low for the vaccinated, fully vaccinated, and the same with deaths going up with the Delta variant emerging in the unvaccinated, while the vaccinated is still being protected against death. And I know this is a busy slide, but it's to show what the breakdown of this result is. Because when Delta was not the major virus, we see that in all age groups, the fully vaccinated had less cases of infection in terms of cases, lab confirmed infection, compared with the fully vaccinated. So the fully vaccinated had less cases of infection compared to the not fully vaccinated. And this was seen for all of the age groups, seen for hospitalization and for death. But when Delta came along, we find that especially in the elderly group over 65s, now there were more individuals in the fully vaccinated population that were getting these breakthrough infections. Again, not so significant in terms of deaths or hospitalizations for the younger people, but for the elderly over 65s, we're seeing a much greater sort of breakthrough infection rate. And what does this mean? And I'm spending a bit of time on this because there's a lot of information out there suggesting that these vaccines are not working now, and maybe they've never worked. Let's go through this carefully. Breakthrough infections causing severe disease are rare in the fully vaccinated. And as of October 18, in the US, 189 million people had been vaccinated, fully vaccinated against COVID. And during that same time, CDC received reports of 41,000 patients with COVID-19 vaccine breakthroughs who were hospitalized or had died. And this represented 0.021% of the vaccinated. So despite what we hear in the media, this percentage, the percentage of individuals being reinfected after being fully vaccinated is low. 41,000 isn't an insignificant number, but compared to the numbers of individuals who have been vaccinated, it represents a very low percent. From the data we've seen, COVID-19 vaccines are effective at preventing severe disease and hospitalization in those under 65 years of age who are fully vaccinated, even while the Delta variant is dominant. And we know that the majority of breakthrough cases are in those that have comorbidities, pre-existing conditions that make them high risk for contracting COVID. These are over 75s and in immunosuppressed individuals who are on cancer treatment or immunosuppressants because they've had organs from other individuals, transplant patients. And about 70% of the breakthrough cases were amongst adults 65 and older, and 87% resulted in death were among the adults 65 or older. So the vaccines are working, they're effective against hospitalization and death, even against Delta, unless you're very old. And we know that the immune response to the vaccine in the most elderly is not as strong as the one that you see in younger people. And also we know that because of the treatments that individuals get for cancer treatment and for transplant individuals, we know that the immune response is lower. There's a lot of Canadian data again to support the same result. And it seems that this breakthrough may be due to lower antibody levels in breakthrough cases versus controls, depending on how you measure the antibodies. But when neutralizing antibodies, see the difference there, peak IgG levels, etc. So it seems that these individuals who are getting breakthrough infections produce lower levels of antibody. But this is ongoing, and these studies need to be reproduced, and we need to follow what's happening. But despite Delta, the fully vaccinated have a 5-fold lower risk of infection, a greater than 10-fold lower risk of hospitalization, and 10-fold lower risk of death. And because of these breakthrough infections, we know that a number of countries are recommending and approving and actually implementing what they call booster, which is a third dose for individuals who are immunosuppressed or who have in the older senior population. And studies that Pfizer performed showed that you could really rebalance the immune response after this third dose to 11-fold increase in terms of the antibody levels, and also increasing the vaccine efficacy back to what was seen after the original 2 doses. So, in fact, Israel is giving the booster to everyone, which may be an overreaction, but most countries are thinking about and implementing the booster for individuals who are in these high-risk groups. We also know that the vaccines have been tested in individuals 12 to 15 years old, and both Pfizer and Moderna have data showing that the vaccine is 100% effective in these groups. And we also know that the vaccines are being developed and tested, and it seems that there's good data in the 5 to 12-year-old group as well. So eventually these vaccines will be available for most age groups. And this is particularly important in terms of school-age kids. Quickly, just to finish, I'll talk about the COVID vaccines. Other COVID vaccines under development. You've probably heard of Novavax, which is developing a protein-based vaccine, and a Canadian company, Medicago, is producing a protein vaccine. Both seem to be very effective in the clinical trials that have been done, and probably will be soon authorized for use. There are inactivated vaccines using technology to kill the whole virus and using that as a vaccine, and also attenuated, sort of weakening the virus, similar to what was done with the polio vaccine. Just to close, I'd just like to remind us of the importance of practicing distancing, hand washing, wearing masks, even if you've been vaccinated and fully vaccinated, because of the data we're seeing now with the Delta. And I'm pretty sure that a lot of the breakthrough infections would not have occurred if the mask mandates, for instance, in Israel, and the opening up hadn't been so rapid after the 2 doses of vaccines were shown to be effective. And I think that we're going to go back to the situation where, until the majority of individuals have been vaccinated, and when there's less chance of variants occurring, we still probably need to wear masks. So as of June 2021, it's estimated that the COVID-19 vaccines have prevented 7.2 million infections and 27,000 deaths in the UK, and in the US have prevented an estimated 279,000 deaths, and up to 1.2 million hospitalizations have been averted. 6.9 billion doses have been administered globally, and 24.9 million doses are administered each year. And so globally, 48.9% of the population, the world population, have received at least 1 dose. But this is the striking piece of information that we need to think about. And it needs to be built into the question of universal booster vaccines, and the whole idea of how can we deal with this pandemic, because only 3.1% of people in low-income countries have received at least 1 dose of the vaccine. Now, my mandate wasn't to talk about safety. I think that's going to come up in a different seminar, but I'll stop there and I'll take your questions.
David, thank you so much. And I'll encourage participants to put their questions in the chat. David, why don't I start with a question about the issue of, I know you didn't get into safety and toxicity, but let's talk a little bit about that. Can you just summarize for us in terms of the time frames when adverse events typically occur following vaccinations?
Okay, that's a really important question. And historically, based on the decades of vaccine development and clinical testing of vaccines that we've all had in the past, historically, we haven't seen vaccine-induced reactions, and I'm talking about really severe reactions, because we know that vaccines can give sort of more immediate reactions at the site of infection, fever, muscle aches, responses that tell us that the immune response is working. We see that quite often, quite a high percentage with all vaccines, including the COVID-19 vaccines. But in terms of severe reactions, these usually occur within 60 days of being vaccinated. And that's why that timeline is a sort of standard time in which a clinical study needs to demonstrate that there are no significant severe adverse events within that time period. Vaccine trials have to show that that time period safety component for all of the studies that are done on vaccines, including the COVID-19 studies. And these were done, and this is really a condition on the regulatory authorities being able to approve a vaccine, which could be totally effective, but it needs to show that there's no adverse events in that safety window. And in fact, this goes on to the question about long-term safety. Vaccines are not drugs that are given chronically, like to treat diabetes or hypertension. They're given at a local site. They're given usually once or twice, and they generally stay at the site of infection. And I would say that 90% of the work of the vaccine is done by the immune system. The vaccine itself doesn't hang around very long. And really, there's been no case of a vaccine-induced severe side effect occurring years after a vaccine has been given. There's been no evidence of that. And it seems that all of the reactions, the severe reactions, the rare but severe reactions, like myocarditis, the blood clots, these have been seen within that 60-day period after the vaccines, the COVID vaccines have been given generally. So again, we're seeing the same profile in terms of safety as and timeline for these vaccines as we've seen in the past.
Oh, great, thank you, David. David, there's been a lot of confusion in the media about efficacy versus effectiveness when we're talking about the vaccines. So can you just summarize for us what we mean by efficacy in the context of the clinical trials, and then effectiveness? You're on mute, David.
Yeah, efficacy is a way of describing how effective a vaccine is based on the results of a clinical study. So in the case of these vaccines, the COVID vaccines, the efficacy was based on performing a randomized placebo-controlled study where some individuals received the vaccine, others didn't, in the context of a phase 3 where there's circulating virus. And the efficacy is based on the results, the ability of the virus or the vaccine, to protect against lab-confirmed disease, occurring, usually looking at 14 days after the second dose and beyond. So this is the efficacy, so it's 90% effective under those conditions. Effectiveness is really the performance of the vaccine in the context of a real-life situation where the vaccines are rolled out to the general population. there isn't a controlled environment where some people, and actually the clinical trials are performed on healthy individuals. So effectiveness is really the performance of the vaccine in real life situation in the community, rolled out to whoever the vaccine has been recommended for.
Great, thank you, David. Now, David, what would you tell someone who feels that the vaccines aren't effective enough to be worthwhile among young children?
Yeah, that's the well. The vaccines, we know the vaccines are effective in young children. I guess you're talking about the 12 to 15-year-olds and younger.
Well, I think the question I was just asking, based on their perception of effectiveness in children versus, in terms of risk versus benefits, I suppose.
Okay, and we know that children, it's rare for young children to be hospitalized or to die of COVID. Although with Delta, we're seeing more and more cases of individuals, but I'm pretty sure it's less than 1% of cases that end up in hospital. But they do get infected. They do get infected. And if you're infected, even asymptomatically, you can transmit virus. You can transmit virus to schoolmates, to teachers, to your grandparents, to the vulnerable individuals who maybe can't get vaccinated. And so, yes, I would say in order to really create what we know as herd immunity, it would be an advantage to protect young children against COVID, not just for themselves, but for the community at large. But it's really an issue of priorities. And I mentioned the fact that very low percentage of individuals in low-income countries have been vaccinated. And so I know there's a debate as to whether we should be shifting the focus to maybe the more vulnerable individuals in developing countries or low-income countries first, rather than, in the West, as we're doing, gradually moving down the age of vaccine recipients to babies, I guess, eventually. So there's some debate about that, but I think scientifically it makes sense to vaccinate children if you can, and if you consider all of the other implications in terms of the world's pandemic.
Yeah, no, that's great. Thank you, David. We're in the last 5 minutes. I just want to remind people to complete the evaluation form. And again, do remember the 2 weeks from now, 2 Thursdays from now, November 11th, we'll have the special workshop that is focused on pediatric vaccines. What can we say about mixing and matching of vaccines, David? What are your thoughts in that regard?
Yes, I mean, I think there's been a lot of confusion about mixing and matching, because initially health authorities, regulatory authorities said that we only are going to use the vaccines in the field in the same way that they've been used and tested in the clinical studies. But gradually, as some vaccines have been had some distribution difficulties, and studies have been done showing that you can, for instance, give AstraZeneca and follow up with a second dose of an mRNA vaccine like Pfizer or Moderna. Results have shown that you can do that safely. And again, you know, scientifically, we've done this, we do this nearly every year anyway. For instance, influenza, there may be half a dozen companies that make influenza vaccine using slightly different technologies. And one year you might get GSK's influenza vaccine, and we know next year the vaccine has to be changed, and it might be a different company that gets the tender. So you might get a vaccine from CSL in Australia, or you might get something from Sanofi. But essentially, these vaccines develop immune responses against the hemagglutinin. And it's the same with these vaccines against COVID. The ones that have been developed now all developed in order to induce a response against the S protein. So they all do the same thing in slightly different ways. And I think the data is showing that it's really possible to mix and match. And especially with AstraZeneca, there have been some issues. It's probably important to be able to use a different vaccine as a follow-up.
How much is cost an issue with respect to, you mentioned the non-industrialized countries, but how important is cost an issue in terms of vaccine availability for those countries?
Oh, it's extremely important. It's very important. And we know that the vectored vaccines, AstraZeneca and Johnson & Johnson, they're less expensive to produce than the RNA vaccines. And in one, I think AstraZeneca are actually distributing their vaccine at zero profit or very low profit at the moment, so that it can be introduced into low-income countries. So it's very important. And, you know, I know that the mRNA vaccine manufacturers maybe think of recouping their development costs, and maybe why the vaccines are so expensive. But I think that they're very simple to make. And I think there needs to be more pressure on those companies to recognize the need to not only depend on countries like the US and Canada to give their excess vaccines to low-income countries, but for them to adjust the pricing so that these vaccines are more affordable.
Okay. David, I want to thank you so much. This has been superb. Thank you so much for this session. And this brings us to the end of today's session presented by Dr. David Burt. Thank you very much. Have a great day.
Thank you.